So I'll tell you now about some of the ways we're trying to, or one of the ways we're trying to get at other potential ways of targeting RAS pathways. And I just first of all consider this point about what we're actually trying to do when we target RAS pathways in a RAS oncogene expressing tumor. So the pathway, we know that a lot of drug components makes it attractive to target these, but there is issue about the level of specificity that one can get for the tumor versus the normal cells, since we know the normal cells are using these pathways as well. The thinking behind why one could go for pathways like this is really that the tumors undergo a lot of damage due to their behavior. In particular, the constitutive oncogene activation tends to mess up a lot of checkpoints, and this causes a lot of damage to the cells. And as a result, in the evolution of the tumor, the cells find ways around that damage, and these are usually relatively small numbers of fixes that the tumor cells get. So they become extremely reliant on a small number of survival pathways, most often ones that are driven by the oncogene itself. And that if one could intervene to target those pathways, you may have a selective advantage on targeting the tumor versus the normal cells, which have probably got a much more complex set of trophic signals. And just as sort of representation of that, one can imagine that a normal cell has a balance of pro-death and pro-life signals with if the cell is around, favoring pro-life. And I've represented those trophic signals as being in a few different colors there, showing they're coming from a few different sources. In the case of the tumor, they're accumulating a lot of damage, largely as a part of the ignoring checkpoints, and they've acquired all these survival signals, but the survival signals are mostly of one flavor. So if you can find a way of inhibiting that flavor, the blue one, which I've just taken out from here, is not affecting the normal cells, but it's going to have a bad effect on those tumor cells. So that, in essence, is this concept of oncogene addiction, which I think underlies a great deal of the thinking in the pharmaceutical industry about how you get at targeting tumors by therapies targeting specific signaling pathways. So we've been interested in this concept and have attempted to look for other ways beyond the familiar pathways in which we might target cells that have dependency on activated RAS oncogenes. And initially we were thinking of this as a KRAS synthetic lethal approach, which is basically that we have a cell. We initially started working with a cell system, which is a colon cancer cell line, HCT116, diploid cell line, and this has one activated KRAS allele and one wild-type KRAS allele. And there's a derivative made many years ago by Shirasawa, in which he deleted the activated mutant allele of KRAS but left the wild-type allele. And these cells reverted to an untransformed phenotype. And so these cells are actually oncogene addicted. The HCT116 cells remain dependent on RAS, the activated RAS allele, and if you take it away, they tend to die. So this is a picture taken from a review by Jeff Settleman, who's thought a lot about the phenomenon of oncogene addiction, and I think Jeff's thoughts on it are that, and which I would largely agree with, that this is really this balance of pro-life and pro-death signals that I showed in the previous slide, with the green being the pro-life signals. And so the oncogene's generating these signals, but when the function is taken away, the signals decay at different rates, with the survival signals decaying faster than the death signals. Now, this is really just a sort of an empirical observation, but I think there is some logic to it in that the damage signals are probably more indirect. They're as a result of the things that the oncogene has done to the cell to change the way it behaves in response to checkpoints and so on. So it probably takes longer for that damage to correct itself than for the survival signals to go away, because they're generally fairly directly controlled by the oncogene itself. Anyway, so it means that in this period of time, the cell is vulnerable to cell death. It also means that these cells, these derivatives, have found a way through that, whether it's just a stochastic thing or that they've selected a subpopulation in some way. So it leads us to be somewhat suspicious of this cell system, but we used it anyway as a starting point. This work really is driven by David Hancock and Michael Steckel, and more recently Miriam Molina in my lab. So the screen, this is the initial screen we did, was on a not a full genome, a druggable genome of 7,500 genes, in which we're looking for siRNAs which will differentially kill the RAS mutant cells, the HTT116 cells, and spare the RAS wild-type cells. And won't go through all the details of it, but basically that comes through with 52 genes that met those criteria. And so I should say this is the assay we're doing here is an apoptosis assay and a cell survival assay. And the main readout we have from it is actually a normalized apoptosis readout. So it's actually the amount of caspase activity divided by the number of cells remaining at the end of the assay. And these are the genes with some idea of how strongly they score, and there's been done in a couple of different systems. There are a few different clones of these RAS-deleted cells. So that gave us a set of genes which seem to have some where these RAS mutant cells seem to show some requirement for those genes, 50 or so. And the question was whether or not any of these could act as sort of surrogates for targeting RAS. If we are still having problems targeting RAS directly, would any of these other things be things that we could target instead of RAS that might be more druggable but have the same effect? One other thing I'll just say about those, which I think is interesting, is that none of those 50 or so genes is differentially expressed in the RAS mutant versus the RAS wild-type cells. So actually these are all things that the cells are dependent on, but RAS is not controlling the expression of them, which was something that surprised me somewhat. I thought quite a few of these things might be actually upregulated by RAS. So to take this forward and to take it away from this specific system, we started looking at other cell systems. And the first sort of disappointment with it was that we realized very quickly that actually the vast majority of the hits from the colon cancer cell line did not show up in any other cell lines we looked at, or even other colon cancer cell lines looked rather similar. And this, I think, has proven to be the case in many of the siRNA screens we've done in all sorts of different areas, that actually there's a huge amount of stuff that's really cell line specific, and it's probably therefore not all that interesting. But by taking a large panel, in this case we used 28 cancer cell lines, mostly lung and colon, and we had half of them were RAS mutant and half were RAS wild type, but otherwise not selected in any way for their genetic status. And we just put these 50 or so genes against those and then just looked for things that looked most like RAS and did a statistical analysis of that. These are the cell lines we used. Probably won't be able to see what they are, but there are many NCI 60 lines and plus some other relatively well characterized ones. And as I said, about 3/4 of them are lung and colon, with a few other from pancreas, over in stomach. And here we're showing, this is the apoptosis, the viability readout, and so clustering the cell lines along the top and the genes down the side. And that's a sort of blow up of a bit, which has the RAS, some RAS controls here marked with yellow. And we get a few things that are looking really quite like RAS. We can also look at the apoptosis data, which is going the other way, obviously, so the red from the more apoptosis is what we want to see, in more selective apoptosis towards the RAS mutant lines. And again, we get a similar sort of analysis. So to put that in a slightly more easy to see format, one can then plot each of these points is a cell line. The red points are wild type for KRAS, and the blue points are mutant for KRAS. And these are the genes that we're targeting. Looking at viability here, so 100 is no effect, and anything below that is killing, and apoptosis where 1 is no effect, and anything above that is killing. And it's probably easiest to see with the viability. But basically what we see so here's what RAS will do to these cells, and so most of these cell lines, most of the RAS mutant cell lines are quite sensitive to having RAS removed. They're mostly oncogene addicted. In fact, they're all oncogene addicted to some extent. And most of the wild type are relatively okay with losing the RAS, although there's always a little bit of an effect. They probably have some dependency on the wild type RAS. And we see a number of things that we've found quite interesting, and in particular, I think these 2 or 3, MMP7, CDC6, and GATA2, which really look very similar to what you get with RAS itself. And with MMP7 in particular, we like because it does very little to the RAS wild-type cells, but does quite a lot to many of the RAS mutant cells. And interestingly here, the ones which don't show much response to this are actually those 3 up there, are all pancreatic lines, whereas the other ones are all the colon and lung, so there seems to be some tissue difference there. These types of screens have been recently reported by others as well, and I think a worry with this type of work is the sort of the discrepancies that people get between, well, they just don't see the same things, and unfortunately this has also turned out to be the case largely for what we've been doing here, and others have reported TPK1 and STK33 as hits in sort of screens of this type, although not done exactly the same way. However, when we look at them by the way we're doing it, in this panel of cell lines, we see no indication that either of those are in any way selective for RAS mutational status, even though we're getting good knockdown. Probably most critically, this is a short-term assay, an siRNA assay, whereas those were reported from longer-term shRNA screens.